Good afternoon, everyone. Myself, Dr. Ranjita. Today, I'm going to discuss a topic that is periodontal pathogenesis. So, just let's start with the topic. So, periodontal diseases is complex in nature. It's multifactorial disease which is largely influenced by genetic, environmental, and microbial factors. So, by definition, periodontal pathogenesis is defined as origin and development of disease. It is a step-by-step process that leads to the development of disease resulting in a series of change in the structure and function. It is a process by which the etiological factor caused the disease. It's given by Carranza in 11th edition. So, histopathology of periodontal diseases. Periodontal pathogenesis is divided into clinically healthy gingival tissues and inflamed periodontal gingival tissues. Even in the gingiva that appears clinically normal, there is a low grade challenge present by subgingival plaque bacteria. Essential mechanism to combat the microbial challenge to prevent bacteria and their products from infiltrating tissues. But an excessive dysregulated immune inflammatory response for a given bacterial challenge leading to increased tissue breakdown. Clinically, healthy gingival tissue is not inflamed, pink in appearance, not swollen, and firmly attached to the underlying tooth bone with minimal bleeding on probing. The dentogingival junction is a unique anatomic feature whose function is a attachment of the gingiva to the tooth. Dentogingival junction is divided into epithelial portion and the connective tissue portion. Both are of fundamental importance in the periodontal pathogenesis. Epithelial structure is then again divided into gingival epithelium, salvular epithelium, and junction. Gingival epithelium. Gingival epithelium appears to be stratified squamous keratinized epithelium continuous with the salvular epithelium at the gingival crest, gingival margin. Covers the gingiva and forms the clinically visible gingival tissues. Covers both the free and attached gingival tissues. Salvular epithelium. Stratified squamous epithelium, non-keratinized, faces the tooth but is not attached to it. Forms the soft tissue lining of the gingival sulcus or the periodontal. Junctional epithelium. Junctional epithelium is unique epithelial structure. It is formed by the co-influence of oral epithelium, specialized for the purpose of attachment of the tooth. Unlike other epithelial tissues, whereas in the body, there is no opportunity for the slugging of the cells from the surface. The extracellular space between the junctional epithelium are also greater than the other epithelial tissue, with intercellular compromises approximately 18 of the volume of the epithelium. This is a result of lower density of desmosomes in the epithelial junctional compared to the gingival epithelium, and hence the junctional epithelium is intrinsically leaky. This has a great relevance in the periodontal pathogenesis, as the widening permit migration of the neutrophils, macrophages from the gingival connective tissue to enter sulcus to phagocyte the bacteria, as well as to ingress of bacterial products as well as the antigens. So, in a low grade inflammation, there is subgingival microflora. Overt clinical signs of gingivitis do not develop because innate and structural defense mechanisms include the following: the maintenance of a intact epithelial barrier, outflow of the gingival clavicular fluid from the sulcus, slugging of the surface epithelial cells of the junctional and the circular epithelium, the presence of the neutrophil and the macrophages in the sulcus, phagocytose bacteria, and antibodies in the gingival clavicular. If the plaque accumulation increases so that these defense mechanisms are overwhelmed, and then inflammation and the classical clinical signs of gingivitis develops. Histopathology of gingivitis and periodontitis. Infiltration of the tissue by numerous different cells, particularly neutrophils, macrophages, plasma cells, and the lymphocytes. Accumulation of defense cells and the extracellular release of their destructive enzymes. There is disruption of the normal anatomy of the connective tissue, resulting in the collagen depletion and subsequent proliferation of the junction epithelium. Now what happens? Vasodilation and increased vascular permeability will lead to increased leakage of the fluids out of the vessels and facilitates the passage of defense cells from the vasculature into the tissue, which appears to be erythematous and edematous. Histological stages of gingivitis and periodontitis. It's given by Page and Schroeder in 1976. So it is divided into initial lesion, early lesion. In initial lesion, the lesion starts within 2 to 4 days. There is no inflammation, and the inflammation can't be evident in microscope also. This lesion corresponds to the histological picture of the clinically healthy gingival tissues, slightly elevated vascular permeability and vasodilatation. GCF flows out of the sulcus leading to the flushing action. Migration of leukocytes, primarily neutrophils, in the small number through the gingival connective tissue to the sulcus. Now, in early lesion, gingivitis is evident clinically. The lesion will develop in about 1 or 4 to 7 days. Week of continued plaque accumulation, there will be increased vascular permeability, vasodilation, and the gingival clavicular fluid flow. Large number of infiltrating leukocytes, mainly neutrophils and lymphocytes. Degeneration of fibroblast, collagen destruction, resulting in collagen depletion areas of the connective tissue. Proliferation of junction and the circular epithelium. So now there is established and advanced lesion. This lesion in established lesion, it will develop in 14 to 21 days at correspondence to the chronic gingivitis. Dense inflammatory cells infiltrate, that is plasma cells, lymphocytes, and neutrophils. Elevated release of matrix metalloproteinases and lysosome content from the neutrophils. Significant collagen depletion and proliferation of the epithelium will also be visible, and formation of pocket epithelium containing large number of neutrophils. In advanced lesion, it marks the transition from gingivitis to periodontitis. Here, the transition will determine the bacterial challenge, post-inflammatory response, and bacterial challenge and susceptibility factors. Predominance inflammatory cells in the connective tissues, predominant immune cells and the plasma cells. The basal lamina differ from other basal lamina that oppose connective tissue, and that type 4 collagen is absent. Inflammatory responses in the periodontium. During the inflammatory response, there are specific molecules which signals tissue damage as the inflammatory response develops. So it is again, this process divides into two parts: subgingival microflora and the host immune inflammatory response. Now, the bacteria are more important because they drive and perpetuate the inflammation, but they are responsible for a relative smaller portion of the tissue damage. It's given by Carranza in 11th edition. So now it comprises of three factors, that is microbial virulence factors, host-derived inflammatory mediators, and the role of specific inflammatory mediators. So, microbial virulence factors, lipopolysaccharide. It contains lipid A component and the polysaccharide component. Lipid component has lipid A. which is found in your outer membrane of the gram-negative bacteria, and it also acts as an endotoxin. It will elicit a strong immune response and have an important role in maintaining the structural integrity of the bacterial cells. Immune system recognizes LPS through toll-like receptors. Toll-like receptors are the cell surface receptors that recognizes microbe-associated molecular pattern, which are conserved molecular structure located in the diverse pathogens. DLR4 recognizes lipopolysaccharide from gram-negative bacteria and functions as a part of a complex of the cell surface molecule, including a CD14 and MD2, known as lymphocyte antigen 96, like lipopolysaccharide component of gram-positive cells was lipoticoic acid, that is LTA, also stimulate immune response but comparatively less potently than your lipopolysaccharide, which is your gram-negative bacteria. Now the CD14 will convert into LPS or LTA, increase production of inflammatory mediators will be there, increased vasodilation, vascular permeability, and recruitment of inflammatory cells by chemotaxis. Now there will be release of pro-inflammatory mediators by leukocyte, which will recruit to the area of the inflammation. So LPS in particular is a key importance in initiating and sustaining inflammatory response in gingival and the periodontal tissues. Bacterial enzymes and the noxious products. Plaque bacteria produces a number of metabolic waste products which contribute directly to the tissue damage. So now the noxious agents are ammonia, hydrogen sulfide, and the short chain fatty acids. These substances have profound effect on the host cells, which are as follows. The short chain fatty acid aid P. gingivalis infection through tissue destruction and also create a nutrient supply to the organism. The bleeding into the periodontal pocket. Plaque bacteria produces proteases and which will capable of breakdowning the structural component of the periodontium such as collagen elastin. These digest proteins and provide nutrition to the bacteria. Now, P. gingivalis produces two types of cysteine proteases known as gingipians. Now, gingipian is again divided into lysine and arginine. So, lysine specific gingipian is RGPA and arginine specific gingipian is RGPB. The gingipians modulate the immune system and disrupt immune inflammatory response, potentially leading to the increased tissue damage. And then they can reduce the concentration of cytokines and digest the inactive TNF alpha, stimulate cytokine secretions via activation of protease activated receptors, thereby stimulating cytokine secretion. Microbial invasion. Periodontal pathogens such as P. gingivalis and Aggregatibacter actinomycetum comitans invade the gingival tissue through your connective tissue. A actinomycetum comitans can invade the epithelial cells and persist intracellular. It's given in the Journal of Periodontal 67. According to some investigators, tissue invasion is a passive translocation process, and according to the others, it is an active process. But the presence of bacteria justify the use of antibiotics to some extent, which are protected from mechanical disruption by the root surface debridement. The bacteria in the tissue represent the reservoir of reinfection after non-surgical management. However, the clinical relevance of bacteria being present in the tissues is better defined. It is inappropriate to make clinical treatment decision, example, whether to use adjunct systemic antibiotics on these premises alone. Now, the fimbria. The fimbria of the certain bacteria Particularly, P. gingivalis play an important role in the pathogenesis. Major fimbrious structure component of P. gingivalis is secretion of interleukin 6, interleukin 8, and tumor necrosis factor alpha. Okay, and then this is the first part. Second part, that stimulation of your nuclear factor kappa beta and interleukin 8 in gingival epithelial cell line. Now the interaction with complement receptor 3 to activate intercellular signaling pathway that inhibit interleukin 12 production. This may be of clinical relevance as interleukin 12 is important in activating natural killer CD8 cytotoxic T cells, which themselves are important in killing P. gingivalis infected host cells such as epithelial cells. Bacterial deoxyribosome and deoxyribonucleic acid. So bacterial DNA stimulates the immune cells via TLR9, which recognizes hypomethylated CpG regimen. Regions of the DNA. These are the regions of the DNA which have cytosine nucleotide is found next to your guanine nucleotide separated by phosphate molecule. Extracellular DNA plays an important role in development and structure of the biofilm formed by the oral bacteria, and their majority of eDNA releases after the cell lysis. This donated DNA is of prime importance as it acts as a meaning of increasing genetic diversity if taken up by the other bacteria, contributing to the antigenic variation and spread of antibiotic resistance, modulation of your host immune response. Hence, the eDNA, extracellular DNA, is a source of genetic information for naturally transformable bacteria in biofilm, and also it acts as a stimulus for the host DNA. So host-derived mediators, inflammatory mediators, are the bystander damage. The inflammatory and the immune process that develops in the periodontal tissue is response to the long-term presence of subgingival biofilm are protective by intent but result in the considerable tissue damage. Majority of the tissue damage in periodontitis derives from the excessive and dysregulated production of a variety of inflammatory mediators and enzymes, which are broadly classified into cytokines, prostaglandins, and matrix metalloproteinases. Now the cytokines. The cytokines play an important fundamental role in the inflammation and are a key inflammatory mediator in periodontal diseases. So cytokines are soluble proteins secreted by cells, which act as a messenger molecule that transmits signals to the other cells and initiate intercellular signaling cascade, resulting in a phenotype change in the cell. So now the cytokines are produced by neutrophils, macrophages, lymphocytes, along with other resident cells including fibroblasts and epithelial cells. Cytokines are again subdivided into profound biological effect and significant overlapping activity. So signal broadcast and amplify immune response is found in your biological effects, mediate connective tissue and alveolar bone destruction through induction of fibroblast and osteoblast production of proteolytic enzyme. Significant overlapping activity. Cytokines functions in complex networks that involve both pro-inflammatory and anti-inflammatory effects that brings together both the aspects of innate and acquired immunity. Role of inflammatory mediators in periodontal diseases. Interleukin-1 family cytokines. Now, interleukin-1 beta has three roles. Key role in inflammation, then innate immune response, and secretion of mediators. Interleukin-1 beta stimulates the synthesis of Pg2, PAF, and nitrous oxide that causes vascular changes associated with inflammation. It increases the expression of ICM on ICM1 on epithelial cells and stimulate the secretions of cytokines. It energizes the other pro-inflammatory cytokines and PGE2 to induce bone resorption. It has a role in adaptive immunity and regulates the development of antigen presenting cells such as dendritic cells. Now, interleukin 1 alpha, intercellular protein and potent bone resorbing factors, and alarmin to signal the immune system during the cell damage. Interleukin 1 RA binds to the interleukin 1 receptor, interleukin R1. However, binding to the interleukin 1 RA does not result in signaling transduction. Therefore, interleukin 1 RA antagonizes the action of interleukin 1 beta. It is important in regulating inflammatory and considered to be an anti-inflammatory cytokine. Interleukin 1-8 interact with interleukin 1 beta. It is produced by stimulated macrophages and monocytes. It results in the pro-inflammatory response, including activation of your neutrophils. It is a chemo attract for T cells and it interact with interleukin 1-2 and interleukin 15 to induce Th1 cells and cell-mediated immunity. TNF alpha is secreted by activation of macrophages, particularly in response to the bacterial lipopolysaccharide pro-inflammatory mediators. Stimulation of endothelial cells to express selectins that facilitates leukocytes recruitment, activation of interleukin 1 beta production, induction of PGE2 by macrophages and fibroblasts. GCF cells of TNF alpha increases as a gingival inflammation develops. So now the anti-inflammatory chemokines, anti-inflammatory cytokines include interleukin 10, TGF beta, interleukin 1 RA, interleukin 1 F5, interleukin 1 F10. Interleukin 10 family of cytokines have multiple pleiotropic effect and possesses immunosuppressive properties. It is produced by TREG cells, monocytes, B cells, and it suppresses the secretion of Th1 cells, Th2 cells, and monocytes and macrophages. TGF beta is a growth factor and cytokine and functions as a cytokine with immune regulatory role. The play inflammatory role in the repair and regeneration, apoptosis, and angiogenesis. Now the chemokines. These are the cytokine-like molecules that are characterized by their chemotactic activity. Chemokines orchestrate leukocyte recruitment in the physiological and pathological conditions and are therefore important in periodontal pathogenesis. The chemokine CXCL8, also known as interleukin 8, is found in the areas of plaque accumulation as well as in the gingival clavicular fluid. Interaction between bacteria and cytokines lead to upregulation of interleukin 8, which is responsible for the development of chemotactic gradient and also stimulates neutrophil migration in the sulcus. Similar chemotactic gradient is present in the periodontal healthy tissues. So now this is the pathway which is of prostaglandins. The cells with the production in the periodontium are macrophages and microblasts. Its levels is increased in the tissues and in GCF at sites undergoing periodontal attachment loss. They are important mediators of inflammation, particularly PGE2, which inflammation and vasodilation and induce cytokine production. COX2 is upregulated by interleukin 1 beta, TNF alpha, and LPS, leading to the increased production of PGE2 in the inflamed tissues. PGE2 results in the induction of MMPs and osteoclastic bone resorption and has a major role in contributing to the tissue destruction that characterizes periodontitis. Matrix metalloproteinases is primarily a family of proteolytic enzymes that degrade extracellular matrix molecules, which is collagen, gelatin, and elastin. It is produced by a variety of cells, including macrophages, neutrophils, fibroblasts, epithelial cells, and osteoclasts. In the latent form, proteases such as cathepsin G produces a neutrophil, and in activated form converts. Now, the MMPs are inhibited by tissue inhibitor matrix metalloproteinases and glycoproteins alpha 1 antitrypsin and alpha 2 macroglobulin tetracycline class of antibodies. So here we have different classification of matrix proteinases, group enzymes and their names. This is the table uh, adapted by Hannah Sayar. And we have groups name gelatinases, stylomycins, matrilomycins, membrane type MMPs and others with their different names starting from MMP1 to MMP20 and from names starting from collagenases to enamelizin. So, in inflamed periodontal tissue, excessive quantities of MMPs, large quantities of inflammatory cells and MMP will convert into inhibitors. Development of collagen depression areas with connective tissues and the predominant MMP in periodontitis, MMP8, MMP9 are secreted by neutrophils and are very effective in degrading type 1 collagen, the most abundant type in the periodontal ligament. Linkage pathogenesis to the clinical signs of the disease. If the inflammation becomes more extensive because of the increase in bacterial challenge, it will lead to increased vascular permeability. Vasodilation leads to the edema and erythema. Gingival swelling, slight deepening of the sulcus, further compromises of the plaque removal, increased infiltration of inflammatory cells will lead to the development of collagen depression and areas below epithelium and epithelium proliferates to maintain its integrity. Now, if the bacterial challenge persists, the cellular and fluid infiltration continues to develop and inflammatory cells soon occupy a significant volume of inflamed gingival tissue. Neutrophils. Neutrophils will divide it into phagocytes and killing bacteria, release a large number of quantities of matrix metalloproteinases, leading to breakdown of the structural component of the periodontium and release potent lysozyme enzymes, cytokines, and ROS causing further tissue damage. And ROS is your reactive oxidative species. The opportunity for repair starts getting limited. The epithelium continues to proliferate apically, deepening the pocket further, which is rapidly colonized by the subgingival bacteria. Now, as the epithelium proliferates, necrosis of the epithelial cells that are more distant from the connective tissue can lead to the intraepithelial clefts and splits. First stage of pocket formation, a cycle of chronic inflammation set in characterized by leukocytes, release of inflammatory mediators and destructive enzymes, connective tissue breakdown, and proliferation of epithelium in the apical direction. Resolution of inflammation. This is an active process that results in the return of the normal hemostasis. Now we have lipoxins, resolvins, and protectins. And lipoxins LXA4, B4, they are the signal macrophages to phagocytose the remnants of apoptic cells at sites of the inflammation. without uh, generating an inflammatory response. They are inhibit neutrophilic inflammatory and transmigration. They inhibit production of inflammatory mediators, protectins. They reduce cytokines expression and also inhibit neutrophilic infiltration. Immune response in periodontal pathogenesis. Immunity is defined as the resistance of the host to pathogens to their toxic products. Innate factors, acute inflammatory cells and chronic inflammatory cells.